Auzubillahi minash-shaytanir-rajeem. Bismillahir-rahmanir-rahim. Dear audience, I am thankful to the Applied Zoological Society of Pakistan for providing the platform of presentation. I am Muhammad Ramzan, MSc graduated, Department of Zoology, University of Education, Lower Mall Campus, Lahore. I am going to present about microbiology and the topic is antibacterial potential of silk extracted from genus Argiope. I acknowledge the efforts and contributions from Dr. Sajida and Professor Dr. Muhammad Arshad from University of Education, Lower Mall Campus, Lahore, and Dr. Hafiz Muhammad Tahir from Government College, University, Lahore. Actually, health issues and food spoilage are major problems of the world due to the bacteria. And WHO has described 12 different families of multi-drug resistance bacteria against which new antibiotics are urgently needed. About 25% annual loss of food throughout the world was recorded due to microbes and important and improper and misuse of broad-spectrum antibiotics cause multi-drug resistance in many bacterial species. The antibiotics discovery was remained so rapid in 20th century, but the progress slowed down later on. Heimer gave the first report about the antimicrobial nature of spider silk in 1988, after which the number of discoveries have been made in this field. Spider silk has antibiotic potential and it is biofriendly and it is ineffective against mammalian cells and no autoimmune response in mammals. In 2011, Wright discovered that antibiotic agents in the spider silk are protein in nature. So the objectives of the present research work was to investigate the antibacterial nature of silk from spiders Argiope and to detect that which solvent is more suitable to dissolve this silk. So the test units to detect this antibacterial behavior of the silk were Escherichia coli, Staphylococcus aureus, Pseudomonas aeruginosa, and Klebsiella pneumoniae. And the treatments were the two types of silk from Argiope spiders. The first one was web silk, and the second one was egg silk. We used 6 different solvents to dissolve these silks from the literature review. These were acetone, 5% sodium hydroxide, ethanol, methanol, distilled water, and urea solution. This urea solution was actually 1:1 mixture of two different solutions that you are looking on the slide. The positive control was tetracycline 30 microgram and negative control was the respective solvents used for the safe dilution. This is a specimen from genus Argiope at Jallow Park, Lahore. So we collect the samples from Jallow Park and Changamanga Forest, southwest of Lahore. We also collect the web of these spiders in clean and uncontaminated F&F tubes, but the egg silk was only harvested in laboratory. Spiders were reared in large clean wooden boxes by providing houseflies as food and humidity and appropriate temperature. Silk was collected from these boxes with the help of sterile rods, and we prepared 3.5% solution of the egg silk and web silk in the described 6 different solvents. Then we heat these solutions at 47 degrees centigrade for an hour and then centrifuge at 7000 rpm for 30 minutes. And resulting supernatant were used as treatments. We prepared the bacteria cultures on nutrient agar media on the plates, and this diffusion method was used for recording the results. 10 microliter liquids from the treatments and negative control was poured on the prepared filter paper discs on the bacteria cultures, and prepared discs of tetracycline 30 microgram was used as positive control. Cultures were incubated at 37 degrees centigrade for 24 hours, at which after which diameters of zones of inhibition were measured as results. SPSS was used for statistical analysis by using MANOVA followed by Tukey's test, and the level of significance was 0.05%. Results for Escherichia coli revealed that the zones of inhibition for positive control was largest and have statistical differences with the negative control and treatments. But the zones of inhibitions of treatments was too small, and these zones were due to the solvents in which these silk were dissolved, as seen through the negative control. If we compare treatments with negative control, it reveals that the effect of treatments were due to the solvents that we use also in the negative control. And these are the results for all the six solvents in which the silk was dissolved: ethanol, urea, acetone, and 5% NaOH in distilled water. These were also the results for Staphylococcus aureus, and same results for Pseudomonas aeruginosa, and same results for Klebsiella pneumoniae. So we concluded from these results that the silk from Argiope spiders have no active ingredient to inhibit the growth of these bacteria: Staphylococcus aureus, Escherichia coli, Pseudomonas aeruginosa, Klebsiella pneumoniae. But at the same time, we observed that 5% NaOH was more effective to dissolve the silk, and we also make further dilutions: 1.5%, 1%, and 0.5% for the web silk in 5% NaOH, and we make these treatments against the four bacterial species that I have already described, and the results were the same. So we recommend that actually a lot of spider species are discovered with silk that have antibiotic nature. These are some citations for in this work. So a lot of work is needed to evaluate the antibiotic nature of silk from different species of spiders, which are still untouched. Further identification and separation of active ingredient in this silk need much work. The production of these biofriendly antibiotics at commercial level may also be possible through genetic engineering and other disciplines. So here are some references. One can follow these references in the citations given in the presentation. And that's all. Thank you very much. Allah Nigeban.